Click for IB Biology. Here we're looking at the polymerase chain reaction known as PCR. There's a PDF, a blank PDF in the description that you can download and annotate with your own notes. There you'll also find links to 10 questions with answers so that you can check your understanding of this content. And then at the end there are channel links to other videos in the DNA replication series, along with an AI prompt that you can use to structure your own research. PCR. PCR, the polymerase chain reaction, was developed by the American scientist Kary Mullis. The purpose of polymerase chain reaction is to amplify a small amount of DNA and to amplify this to a larger sample so that you can carry out various tests associated with DNA technologies. Some of us will have been the beneficiary of PCR in recent COVID-19 tests. The technique relies upon having our original DNA sample and on the action of DNA polymerase enzymes, which we have previously studied. In this case, however, we are going to be working with what is called Tac Polymerase. Now, Tac Polymerase comes from Thermus aquaticus, which is a bacterium that lives in hot springs, and as a consequence of this, the enzymes that this species of bacteria have are thermostable at very high temperatures, such as the 95 degrees, which is used in the PCR chain reaction mechanism. So we take advantage of DNA polymerase, but it's actually from the Tac DNA polymerase, Tac polymerase. You would also require a supply of the nucleotides adenine, thymine, guanine, and cytosine. And initially we begin by applying a high temperature that causes the strands of the DNA, the two polynucleotide chains, the hydrogen bonds between the base pairs break, and we have the separation of the strands. At that point, we add primers to the strands and then cool to 40 degrees centigrade. The primers on cooling anneal, that is, that they fix to the template strands which have been created in the previous heating cycle. With the adenine, thymine, guanine, and cytosine available, complementary base pairing takes place along each of the strands, and when heated again to 72 degrees centigrade, the strands begin to DNA replicate. So our original one strand has now become two strands. So we've begun the amplification cycle. The cycle that we've just outlined, which took us from one DNA molecule to two DNA molecules, was a single cycle. And then the amplification then carries on, where two DNA molecules will then go through the same heating and cooling cycles to produce 4, then 8, 16, and so on through many, many cycles, where 20 cycles will then provide us from the original one DNA molecule, we would end up with excess of 1 million DNA molecules. And so we have amplified our sample of DNA. This amplification is essential, as often the source DNA that we're working with is in low quantities, but the tests that we wish to carry out on our DNA will require large quantities of DNA. So PCR is a way of amplifying a small sample of DNA into a large sample of DNA for various tests that we will look at in the next video, PCR. Links to some of the tests that we can use with PCR are appearing in front of you, also to the technique of gel electrophoresis, and of course links to the channel and the playlist.